The topic of this discussion is character mapping. You're already familiar with this, and some of this is going to be review, and a little bit of it is kind of to tie all of the things we've talked about with regards to character mapping together. So, when we're talking about a character, remember, in this class we're dealing primarily with discrete nucleotide data, that is DNA data that's been aligned so that we can compare homologous parts of that and make some assumptions about relationships, and in this case with mapping ancestral states. But we can also apply this principle to non-molecular characteristics. So if we wanted to look at the history of let's say a physical feature like a bone, we could look at bones of modern species, map their patterns onto a modern phylogeny, and that would help us say something about the ancestor. Now, prior to phylogenies and mapping of characters on phylogenies, the only way that we had to understand things about ancestral states, about past conditions, was the fossil record. And there were some non-parsimony, pretty obvious conclusions. So for instance, once you understand that all mammals share a common ancestor, it's easy to see that if there's a characteristic that all mammals share, such as producing milk for their young, that the ancestor must have had that also. But other characters are less obvious. And so as we map things onto a phylogeny, we have an explicit statement about the way that ancestors were, even for complex characters. But there are some limitations and drawbacks to this. So let's review, and I'm going to look at, as we go through this, we'll go back and forth between the PowerPoint and a document that I can draw on here that's pulled from the third slide, I think, or the second slide here in the PowerPoint. So you should be familiar with these different types of character patterns. They're pretty straightforward, right? A synapomorphy. So let's look, and we'll look at some examples of synapomorphy. Right, so I've got a character matrix here, and I've got two competing phylogenies, and again, we would map all of them on there and see which one was the shortest if we were interested in finding which of these is best based on a parsimony criterion. That should be reviewed for you, should be familiar. Let's look at the third character here. Everything has an adenine except for species E and F, which have guanines. Now, the most likely explanation for that, again, this is the parsimony criterion, is that somewhere in the ancestor of E and F, let's make that dark so that we can see it. How's that? Somewhere in the ancestor of E and F, there was a change, and that would be the same for both trees. That character doesn't help us distinguish between the trees, and that's a fairly unambiguous marking. Now, again, that's just a single character. It's not 100%, but it's the most likely. It's possible that perhaps this character evolved once in the ancestor here and then was lost in that branch. That's less likely, and so we don't like it as much, and overall we just assume that, although we might be wrong occasionally, that if we choose the most likely character mapping for all of the characters we have, that we'll find the very best overall sets of relationships, right? So we'll go back to our single most parsimonious marking. We'll look just at this tree on the left. Let's look at character number 4. It's a little bit more complex because there are 3 different character states for that character. We have species, the outgroup, and A and B all share a adenine, where species B and C share a thymine, and E, F, and G share a cytosine. So we'll just do one character at a time so it doesn't get too busy here. So for character number 4, this is what we would do. Species B and C, there'd be a mutation there. That's why they both have a thymine. We do the same thing on this tree. And then we've got this interesting one here where it says E, F, and G all have a cytosine. And again, we could do that both on both trees, E, F, and G, and that's easy. And now notice that as we put those marks representing evolutionary changes on here, we've made explicit assumptions about what the ancestors are. And so we would assume the ancestor of B and C, because of that change somewhere along there, had a thymine at that position. Put a little T there. We would assume that the ancestor of E, F, and G, because we assume the change there, had a cytosine at that position. And again, because those represented changes, we would assume that all of these other ancestors must have had adenines at that position. So we have explicitly inferred ancestral states. Now again, this is a bit of a review. Hopefully it's not much. And realize we can do that for every single position on the phylogeny. And it's pretty straightforward. It's easy. We're making some very simple assumptions. But again, you want to be a little careful because parsimony says this is the best way to do it, and it's not 100% guarantee that that's the way it occurred. And so sometimes in the past people say, oh, it's 100% guaranteed that that was a C. And that's not exactly what parsimony is saying. It's just saying that that is the simplest explanation. And so averaged out over all of the character states, that helps us find the phylogeny. But it gives us a very unambiguous marking if we do it with a parsimony mapping. Now, there are instances where we might have a character that doesn't give an unambiguous marking. And so you can practice and go over all of these. I'm going to add a character. I think it would be character number 18. Let's add one just because I don't have one in this example. Let's add one that's a little less straightforward. It is ambiguous. So let's assume, and just for simplicity's sake, since we start with an A, let's put an A in here for every single species, okay, until we get down to the F and G. And so let's say F and G share a thymine at that position, and all the others have adenines. And this would be the same on either tree, so I'm only going to mark it on, we'll just do the one on the left here. So if F and G have a thymine, you might be tempted to say, oh, look, it's convergent. We're going to mark two separate changes because we don't have a single common ancestor for just F and G. F is more closely related to species E than it is to species G. And then you would just might be tempted to say, oh, I'm done with it. We mark it as convergence. That's easy. And that's true. That's one way to do it. But there's another way to do it in a strict parsimony framework. We could assume that that mutation to a thymine happened in the ancestor, and then there was a back mutation in the branch leading to species E. And that would explain why F and G have a thymine at that position. It's an ancestral one, and this is a simplesiomorphy for that group. Happened in the ancestor, but then was lost in one of the descendants. Okay, so in this case, there's not a single unambiguous marking for a parsimony framework if we're going to be a very, very strict, go by a very, very strict parsimony where everything is weighted equally. But if we have some information about mutation, so realize that this mutation represents a mutation from an A turning into a T. Whereas this mutation is slightly different, right? It is a T mutating into an A, into an adenine. And so if we assume that all of those changes are identical, then it doesn't matter whether we mark it as a convergence or a simplesiomorphy. If, however, we think that a T mutating into an A is a little bit more likely, and maybe we do that based on observations of the data and a preliminary tree with a neighbor that's made from a neighbor joining tree. There are a couple of things we've talked about that might help us to do that. But if we assume that that was more likely, and again, we're kind of talking about models of evolution now, we would actually prefer the simplesiomorphy instead of the convergence, because here we have two characters with one that's slightly more likely than the other, whereas back here with the convergence, oops, let's mark it on there again. Back here with the convergence, I have to mark two separate adenines mutating into a thymine. And again, if the reverse is slightly more likely or slightly more possible than this one, then I prefer one of these and a reversal rather than two of these, which are slightly less probable. Okay, so people begin to realize that if we know something about changes, then a strict parsimony approach is probably not the best way to do it. Now, one last thing before we move on here. So if you want, you can practice. So we should be able to recognize a synapomorphy, a simplesiomorphy. An autapomorphy is simply a character that's only in one species, and I don't have any autapomorphies in this group. But for instance, if everything was an A, except for, let's say, species G had a C. So A's all the way down, and species G has a C or a cytosine at that position. We can always just mark that here on the branch leading to G. It just tells us G is different than everything else, which we already knew. That's why we're defining it as a species. And so an autapomorphy is a derived characteristic that only one species has. And so those are really not informative for parsimony relationships when we're trying to figure out the best phylogeny. Okay, but the last thing I do want to do, let's get rid of all of this, and I'm going to bring in an image from a more complex character. Okay, and this is one we already talked about. You can go back and review that video if you need, if you're having trouble with this, but this is just one more run through it. It will help you to review it, go over it, hopefully cement it in your head. Okay, so let's pull this in here where I can draw on it. Just a bit smaller. Okay, and let's look at this more complex character. Okay, now to make it a little easier, I'm going to write the DNA base pair above each of the species, so I just remember what they are. So adenine, cytosine, cytosine, adenine, guanine, thymine, guanine, and adenine. And you look at it, and immediately you're not going to be able, probably, unless maybe you're very good at this, I don't know. I can't look at that and immediately say, oh, I can map a change here and here and put all the most parsimonious way to map it, because it's much more complex, right? And that was the fourth category where we looked at, where we have a more complex pattern than simply a synapomorphy or a simplesiomorphy. And this is common in DNA data, particularly when you have a high rate of evolution, lots of different character states. So we're going to quickly do this. I've already done it in a previous video. In the down pass, we assign tentative states for the ancestors that are going to be ambiguous. So I've got to vote for a T, a G, and an A for that one. But notice when I come down to this character, I've got a T, a G, and an A from the node above. And from that branch above, I have another vote for G. So I assign that to be an ancestral state of a G. Now I come down here, and I do that one down pass, a C or an A. Because we have a vote for C from that branch, a C and an A from the node above it, I'm just going to assign that the unambiguous one of a C. Now notice this one is an example where we have an ambiguous one all the way down to the base. And this shows you some of the weaknesses of these parsimony, even though they could be explicit. So I'm sorry, I made a mistake there. I'm going to get rid of the A. It should be just a C from the node above and a G from here. And then we would put a C, G, A there. And again, under a strict parsimony framework, what I'm saying is, because I don't have any other information down here, I'm just saying that, oh, I don't know if that's a C, a G, or an A. It could be any of them, and then depending on which it is, it's going to change how I map characters. But there are several most parsimonious ways to map this. Now, for illustration purposes, let's say we got one more outgroup, so we'll call that outgroup number 2. And that outgroup number 2 had an adenine. So if that was the case, now we have a CGA, an A from there. It's very easy to say that ancestor had an A under this framework. Now we're going to change that to an A, but we still are left at this place where, oh, I can't say anything because I had an A in my up pass. The information from underneath says adenine, but it only says a C or G here. So if we go back even farther and assume that O2 instead was a cytosine, that gives us a little bit of a different outcome. So we would say cytosine up here, CGA. We're going to vote for a cytosine there, right? And now we can begin to make these explicit statements about all of the ancestral states. So that would become a cytosine. That would become a cytosine. Now I'm doing my up pass, and again, review. This one, we just look at the node beneath it, cytosine. So that C and A gets turned into a cytosine. Now that's already been assigned, so we don't even need to use the information underneath it, but this one has not, so we use the information from directly beneath it. We assign that to be a guanine, and now that it's a guanine, we can use it to assign this to a state of a guanine. It's a little bit messy here, but and now that we know all of the ancestral states, we can easily map in where changes occurred. There must have been a mutation along that branch because we have a C here and a G there. There must have been a mutation along that branch and along that branch. And then there must have been a mutation here because that, oh, sorry, it's the wrong place. Ancestral C, so the mutation would be there. So we've only marked 3 changes on this phylogeny, 4 changes, 1, 2, 3, 4. That's guaranteed to be the most parsimonious way. And in this case, that's an unambiguous marking for this methodology, this parsimony. But it doesn't guarantee to give you a single unambiguous marking. So there are some problems with it. Okay, so hopefully you're familiar with this. Now, there is a form of parsimony that's basically an extreme force of weight form of weighted parsimony, and it's called Dolo Parsimony. And in Dolo Parsimony, we don't treat everything equally, which is what we do under his. Fitch parsimony is basically just equal weights, where we can have changes and mutations back that are equally likely. And so here we've marked 2 changes. You could call it 3 if you want to say, well, it had to have happened somewhere in the ancestor. Okay. In the Dolo parsimony, we assume that it can only happen once. So here we're assuming that this black character state evolved once in the ancestor, was lost, and then evolved again. And that is would assume 3 mappings. But in Dolo parsimony, we assume that once a character evolves, it's never going to be re-evolved. So we aren't allowed this state here where it evolves once and then evolves again. And so here we would actually have to map a loss of a character on there and we'll just show you here so you know where it would be. We'll put a little circle there since that's what they used. Right, we'd have to map the loss of this character shape or character state in multiple places. But again, because we're assuming that losses are much more likely to occur than gains, that's the best way to do it. Okay, so even though we say, oh, in this tree here, I've had to map 1, 2, 3, 4, 5 different events, we like it better than this one, which only has 3 events, because we're assuming that a regain, a re-evolution of a feature is very, very, very unlikely. Okay, so when we apply this to physical characteristics, this makes a lot of sense. So these are all insects that have ancestors that had wings, but none of them have wings at any stage of their life cycle. And most of these evolved naturally, although I even put an artificial example. This is a Drosophila melanogaster mutant, which we use in the lab quite frequently, that doesn't ever grow wings at all. Now that's pretty obvious. We compare it to its ancestors. It is a very obvious recent loss. These others are a little less obvious in some cases, but they're all losses of wings. And so what we would do is we would map the origin of wings at the base here of Neoptera, which is the technical term for the insects that can fly. Sorry, Pterygoda is the technical term, and I think I need to go one more branch down. So Pterygoda is the technical term for all insects that can fly. Neoptera is a subgroup within that. That's not important. But we mark the origin of flight here. And then if we had to map the loss of flight on this phylogeny, we would actually be putting it in dozens and dozens of places. So it turns out that loss of certain features, particularly if they're exposed to similar environments, and we see this commonly. So very commonly, parasites lose their wings. We also have other pressures, right? If we're living in an area where there are lots of birds that are taking insects on the wing, that might be a pressure, and insects that lose their wing. It's a common thing that can happen by many genetic mechanisms for organisms to lose a structure. And so we might mark the loss of that on there in many, many places, rather than assuming that wings evolved independently many times. And that makes a lot of sense in the context of these complex morphological characters. So Dolo parsimony was originally an extreme form of weighted parsimony that was developed for these complex morphological characters. But how does that apply in terms of DNA, where we don't really have, at least not in the same sense, this idea that some things are not going to be re-evolving over and over again, because we're just looking at very simple mutations. And even though one might be a little bit more likely than the other, it's not this very, very extreme form that we might try to impose on our characters with Dolo parsimony. And so we came about, and you guys recognize this in previous discussions, with this idea that we might want to give some characters different weights because of what we observe in our data. But putting that in parsimony and implementing that in parsimony can be different. So at DNA, we can gather these large data sets, map them onto a pretty decent phylogeny, like a neighbor joining phylogeny, and come up with some idea about how often certain traits come. And they're estimates, so they're subject to some error, but they're useful, and they are a lot better than just simply treating everything either equally or giving some arbitrary weight to different character states. So at the level of DNA, we gather large data sets. We implement those patterns that we're seeing into our models of evolution, and that helps us not only with finding the best tree overall, like in a maximum likelihood or in a Bayesian framework, but it also gives us a new methodology for mapping trees, or for mapping characters onto trees, sorry. So let's look at that just very quickly. And we're not going to get into all the details of this, but I'm just going to highlight some of the main differences that you need to know. So we can map states not only in a parsimony framework. So in this example, all of the black ones share one, I think it's a weighted a food source. They all eat one thing. The gray one eats a second thing, and then the white ones all eat a third thing. So I have different foods. And obviously, this is going to be tricky from the fossil record, although maybe if you had good preservation of teeth, you could infer something about their diet, although that's not always the case. So this is something where we may not have, even if we have a good fossil record for this group, we may not be able to say much at all about their very, very specific diet. Maybe only we can say things generally. So it's useful to map them onto phylogeny. And it's a pretty unambiguous marking in parsimony. We would map the origin of this diet preference at the ancestor here. This changed the diet preference there, and it's obvious that the ancestors had this, whatever the white one represents, that's the food that they would have eaten. And again, fairly straightforward, unambiguous, but realize it doesn't show us that we might be uncertain about some of these. So in a maximum likelihood, there are two main differences. Number one, the branch lengths are informative. So very, very long branch lengths might give us more information about whether a character evolved along that branch. As opposed to it being a very short one, then it's unlikely for those characters to evolve. So in the analysis, we incorporate in branch lengths as part of our ancestral state reconstruction. Okay? And then also, we represent ancestral states as probabilities. Now, the probability can be 100, or at least very, very close to it, like we've done here in these ancestors. But a probability might be, 74 or 5 here, or it might, the probability of this one maybe is only like 15 to 20%. And so the two main differences with maximum likelihood character state mapping is that we incorporate the information from branch lengths, and then we have probabilities on our ancestral states rather than just saying, oh, it's either this one or this one. So it's a little bit less satisfying maybe for some people, but probably more realistic. Okay, the last thing that I want to talk about when we're talking about character states is, so far we have assumed discrete character states, DNA or even morphological characteristics. They either have wings or they don't. But many cases, and maybe even wings, because there are some insects that have brachypterous wings, wings that are still there but not large enough to fly, or that can barely help them fly. So maybe it's more realistic to treat some characters as non-discrete or continuous traits. So for instance, height in humans, right? That's a continuous trait where you can be tall and short and everything in between. It's not either or. And so here we have numbers representing some sort of continuous trait. And this is somewhat discrete because it looks like you're only allowed to be at the integers. But assuming that maybe you could be in between 2.45, 6.34, we come up with methods in a parsimony and also in a Bayesian framework. And both there are methods to do this that we're not going to get into, but where we could come up with some intermediate trait that is the most likely based on what's above it and what we put below it, that this would be the most likely. And so then we could infer rates of evolution along these branches and also some sort of a ballpark, and maybe even with ranges of probabilities, but a ballpark of what the ancestor was, or any ancestor was, based on these states in between. So just be aware that you can map non-discrete continuous, even continuous traits onto a phylogeny and have some estimates, and that works in both a parsimony framework and a maximum likelihood framework, although we're not going to look at the details of how those are done. So just to wrap up, mapping character states is very, very critical for our understanding of ancestors. And if we had a character state, we could even reconstruct in your paper, the Cunningham paper for this lecture goes into that and says we could maybe even reconstruct an extinct, say, amino acid sequence to represent an extinct protein. And therefore, we could say something about that and maybe even test it in the lab, because we can artificially make proteins and test to see if they would have any physical differences. And so it gives us an extra insight and extra tools, very powerful tools, for understanding the history and evolution of things across a phylogeny. However, recognize to do this, we need to start with a phylogeny. So it all goes back to having good data with a good alignment, using maybe even multiple methods, seeing if they're congruent, and then using that well-established, well-supported phylogeny to then map individual characters on it. And even if that individual character doesn't support the phylogeny, if we have many, many other characters that do, then suddenly we can say something about that one character, which might be abnormal, might have evolved in a very complex manner with high rates of evolution or back mutations or many other things. That one character mapped onto the well-supported phylogeny will give us great insight into the evolution of that organism.